dear students, welcome to this series of lectures in botany, and today our topic is evolution of seed habit in plants. Seeds represent the generative propagules of plants that hold key to their successful life cycle and better survival in diverse habitat conditions. Besides, seeds also facilitate plant transport at different spatial scales, thereby causing their transport to long distance as well as short distance as well. Seeds also offer protection to plants and help them withstand and overcome harsh conditions through dormancy. In fact, the evolution of seeds and seed habit represents a remarkable life history transition for spermatophytic plants due to a number of reasons to be discussed subsequently. Since their first appearance in the late Devonian, seed plants have come to dominate almost every terrestrial ecosystem, encompassing a greater range of habit and habitat than any other group of tracheophytes. The evolutionary success and ecological diversity of this group generally stems from their reproductive system involving seeds, which facilitates these plants to exploit habitats not accessible to most lower vascular plants. So, the objective of the present lecture is to give you an opportunity to learn about the various aspects of evolution of the seed habit in plants. By the end of this lecture, I hope that you will be able to answer some of the fundamental and interesting questions related to the topic, such as how and why seeds and the seed habit evolved, how natural selection has operated to favor the seed life history phenomenon, and of course, what advantages do seed habit confer to the plants. So, for the question how and why seeds and seed habit evolved is concerned, from the Darwinian perspective, seed habit is considered to be a pre-adaptation for the quick dominance of the angiosperms. In addition, the enhanced sporophyte, reduced gametophyte, and a wide range of morphological adaptations such as roots, leaves, the cuticle and stomata, etc., allow seed plants to occur in a wide variety of habitats and dominate the terrestrial flora of Earth. The seed habit per se, in addition to vegetative traits such as production of wood by secondary meristem, cambium, contributed decisively to the evolutionary success of the gymnosperms and angiosperms. Evolution of seed habit has at least two important but independent aspects. First is the evolution of life history, what we call the seed habit, that is the delivery of a male gametophyte to a female gametophyte that is fixed on the parent sporophyte. Second aspect is the evolution of ovular, particularly integumentary morphology. The seed habit life history, in contrast to seed morphology, differs from free sporing heterospory in just one fundamental way, that is, by the delivery of the male gametophyte directly to the female gametophyte through pollination. This bypasses the free release of female gametes into the environment, and thus the environmental limitations placed on the free sporing system by the water mediated fertilization. In fact, pollination by its nature places plants with this life history in an entirely different ecological and hence selective context than lower vascular plants. It constitutes a fundamental phenotypic breakthrough that opens to these plants a vast resource pool, the terrestrial landscape previously unexploited or poorly exploited by sexually reproducing plants. And so for the second important question is concerned, how natural selection has operated to favor the seed life history phenomenon. The seed habit, let me tell you, is the most complex and successful method of sexual reproduction in vascular plants. The seed plants, what we also call as Spermatophyta, generally comprise two major groups. The acrogymnospermi, also referred to as gymnosperms, with a conservative estimate of about 800 living species, and the angiospermi, also referred to as angiosperms, with a conservative estimate of more than 250,000 living species. Before we proceed ahead, let us also dissect some frequently used terms in the seed biology. Number 1, ovule. It refers to the structure that consists of the integuments surrounding the nucellus, the megasporangium, or in other words, unfertilized immature seed precursor it is. Second, nucellus. It refers to the megasporangium, which surrounds the gametophyte and can develop into perisperm after fertilization. Megametophyte. It refers to the female gametophyte, which contains the female haploid egg cells or gametes, and several thousand of these are in gymnosperms, or typically 3 to 8 of these are in angiosperms. The integuments, which refer to 1, as we see in case of gymnosperms, or 2, as we see in case of angiosperms, outer layers of the ovule, having an apical opening, what we call as micropyle, which develops after fertilization into the seed coat, also called as testa. Another term is the micropyle, which is the apical opening of the integuments, which allows the pollen tube to enter the nucellus to release sperm for fertilization. Testa refer to the seed coat derived from the integuments of the ovule, dead maternal tissue in other words. One more term is the endosperm, which arises from the fusion of the second sperm nucleus with the central cell nucleus of the embryo sac during double fertilization. It's a nutritional tissue during seed development, which has an important role, and in the mature seed of most of the angiosperms, it is in between testa and embryo. Perisperm is another term which is derived from which refers to a structure which is derived from the nucellus after fertilization, maternal nutritional tissue in the mature seed of some angiosperms. Ovary refers usually to the lower portion of the angiosperm pistil, carpel or fused carpels containing ovules. Fruits are mature ovaries, and ovary tissue develops into the pericarp. And pericarp, you must know, is the fruit coat of the angiosperm fruits, which develops from the mature ovary wall and other flower tissues surrounding the seed. These groups are by far the most diverse lineages within the vascular plants. And before we proceed ahead, we need to understand the major characters which are typical to the modern seed plants. Some of these include: number 1, the megasporangium, the nucellus, contains a single functional megaspore. Second, the megaspore is usually one member of a linear tetrad formed by the meiotic division of a single megaspore mother cell. Third, the megaspore is retained within the megasporangium. Fourth, the megasporangium is enclosed by a sporophytic structure known as the integument, and the megaspore axene is thin or sometimes absent even. Then also the megaspore and megasporangium are united histologically, and one more important character is that female gametophyte develops while retained and nourished by the maternal sporophyte, a very important character. And microspores, pollen grains, are captured before dispersal of the megasporangium. One more thing is fertilization and formation of an embryo usually occurs before dispersal of the megasporangium. Then resources are only committed to pollinated ovule or to the ovules that have been fertilized. And of course, one more and very important feature is that microspores germinate distally to form pollen tubes that grow through the sporophytic tissue to reach the female gametophyte. The challenging issue for you is, however, to know why these characters evolved and how they are interrelated. Let me give you a brief idea about the plant origins. Seed plants appeared more than 350 million years before with the origin of gymnosperms, and flowering plants, I mean angiosperms, the other surviving lineage, appeared 200 million years after date, I mean 200 million years later. But the rapid rise and early diversification of angiosperms during the Cretaceous period has been described by Charles Darwin as an abominable mystery. If you see the sequence of events from bryophytes to angiosperms, you will see that in bryophytes, gametophyte is dominant, but sporophyte is dependent and gametophyte is independent. In seedless vascular plants, sporophyte is dominant and sporophyte initially is dependent, but gametophyte is independent. And in seed plants, sporophyte is dominant and sporophyte is independent as well, but gametophyte is dependent and microscopic, very small. And we have two groups of seed plants, gymnosperms and angiosperms. In gymnosperms, gametophytes develop inside cones, while as in angiosperms, gametophytes develop inside the flowers. There were three major reproductive adaptations in the evolution of the seed plants, very important for you to understand. Number one, reduction of gametophyte, that means gametophytes of seed plants are microscopic, so they can exist within the sporophyte. Second, seeds became an important means of dispersal. And third, pollen or sperm cells eliminated the liquid water requirement for fertilization. Let's very briefly understand the implications of these reproductive adaptations. But before that, you need to keep in mind that all seed plants are heterosporous. Female spores give rise to the female gametes and male spores give rise to male gametes. The megasporangia produce megaspores, I mean the female, and the microsporangia produce microspores, I mean the male spores. And the megasporangium is enveloped by a layer of tissues called integument, and the whole structure, I mean the integuments plus the megasporangium, is called the ovule. So for the first genate is concerned, that means the reduction of gametophyte, it continued with the evolution of the seed plants. In seed plants, the very small female, I mean the haploid 1n gametophyte, develops from spores retained in the sporangia of the diploid, I mean 2n sporophyte. The question is, why so? The important reasons are, very delicate female gametophytes are protected from the environmental stress. One. Second, embryos that are produced are also protected from stress. And so for the second genate is concerned, seeds became an important means of dispersal. Seeds are, resistant to environmental stress and are multicellular, complex structures that consist of a sporophyte embryo, a food supply and a protective coat, of course. And the embryo develops from the fertilized egg, which is derived from a spore retained within the sporangium. The sperm derived from a spore is carried or produced by a pollen grain released from the sporangium. And the third very important tenet is that pollen or sperm cells eliminated the liquid water requirement for fertilization, and this facilitated the transition of plants from aquatic to terrestrial habitats and their subsequent diversification thereon. Having talked about this, very important aspect of seed evolution is the evolution of dormancy. You know, seed dormancy can be defined as an intrinsic block to the completion of germination of a viable seed under favorable conditions for germination. For example, temperature, humidity, light of the corresponding non-dormant seed. And seed dormancy controls germination timing in response to the seasons and plays an important role in seed plant evolution and adaptation to climatic changes. Germination timing may strongly influence the rate at which species can expand their range and may play a very important role in determining the survival and extinction also during the climate change. And these seed dormancies of different types, and very recently in 2004 Baskin and Baskin in fact proposed a comprehensive ecophysiological classification system which includes 5 classes for the whole seed dormancy, ranging from the morphological dormancy, which is regarded as the ancestral one, followed by more advanced physiological dormancy, morphophysiological dormancy, physical dormancy, and also combinational dormancy. This classification is mainly based on embryo to seed ratio, ES ratio, which describes the relative size of the embryo within the seed. A high ES or embryo to seed ratio, for example, or 0.9 means that embryo fills up most of the seed volume, whereas a low ES ratio, for example 0.1, means that the embryo is tiny and the nutrient storage tissues, that means endosperm, perisperm, megametophyte, they fill up most of the seed volume. Studies have shown that ES ratios have increased in derived angiosperms compared with the ancestral angiosperms. Thus, there seems to be a general trend of increasing relative embryo size during the evolution. That means higher ES ratio for both angiosperms and gymnosperms. Based on this, it has been proposed that morphological dormancy is the ancestral dormancy type amongst the gymnosperms and angiosperms, and the evolution of larger embryo size likely resulted in occurrence of non-dormant seeds because the embryo did not need to grow before germination. It is therefore thought that increased relative embryo size is one of the main determinants or requirements for the evolution of other classes of seed dormancy. One more aspect that we should not lose sight of is the seed size evolution. Let me give you a very brief idea about this aspect as well. In view of the centrality of the seed size or seed mass to many aspects of plant ecology and evolution, let's discuss it very briefly vis-a-vis evolution of the seed habit. During a period of rapid angiosperm diversification, around 85 to 65 million years before, angiosperms moved out of the tropics and shifted from being predominantly small seeded to having a much wider range of seed sizes. Let you understand that extant angiosperms have seed masses spanning more than 11 orders of magnitude, from the lint-like seeds of orchids up to the 20 kg seeds of the double coconut. Gymnosperms have somewhat less variation in seed mass, but have usually larger seeds than the average angiosperms. Plant size has been found to be the strongest correlate with the seed mass across a diverse assemblage of plant species, even stronger than mode of dispersal or environmental conditions. It has been suggested that species with a large seeds or seed mass have an advantage under low light conditions when their greater protein and lipid reserves or their more advanced development can facilitate growth favorably. However, you must note that large seeds usually come at the cost of seed number per flower or fruit. In addition, large seeds cannot be physically borne on small plants because of the weight of the seed, which may partly explain the association between plant size and seed size, of course. Besides, the plant size or seed size correlation is that there are common genetic components that determine the seed size, plant size and the size of the other plant organs. And recently, attempts have been made to correlate genome size with the seed size. So, dear students, let us conclude the fascinating story of the origin and evolution of the seed habit that started in the late Devonian around 370 million years ago, with the note that three major evolutionary trends were important for the transition from progymnosperms to the seed plants. Number one, the evolution from the homospory to heterospory. Second, the evolution of the integument, and third, the evolution of the pollen receiving structures. This includes the transition to water independence of the pollination process. Finally, we discussed the range of variation in seed dormancy and seed size or seed mass and their ecological correlates. Having said all this, it is important to end the lecture with the fact that there are yet a series of unanswered questions that are ripe for further research in the field of seed biology, which I hope some of you may be able to answer through your research. So with this, I take leave of you as of now until I meet you next. Goodbye.